Welcome back. Continuing our discussion from multi-photon microscopy, I think it will be apt to close this chapter, close this module by talking about multi-photon nanosurgery. If you can send multiple photons and then you can look into the a specific emitted photon out of it and try to image it, what will happen if a high intensity laser beam of similar multi-photons, 2 photon, 3 photon together are sent at a specific area which you have previously imaged, and you think that that needs to be eliminated, surgically taken out, surgical strike if you say, at the specific area. Why can't we do that? So, that is today's topic. We discuss about multi-photon nanosurgery. So, think how far or how advanced our knowledge and our technology is going, where non-invasively by sending laser light we are attacking specific areas of the brain, few neurons at a time, either removing it altogether or we are modifying it, right. So, what is multi-photon nanosurgery? Multi-photon nanosurgery is a cutting edge technique that utilizes the principle of nonlinear optics and precision laser manipulation to perform surgical operations at a nanoscale level. You are sending laser light, highly focused laser light of resolution few microns at specific areas of the brains and perform surgical operations in living biological samples. This is important. You are not simply taking the portion out, putting it in a slide and then trying to do it. You practice using that, but the real deal is when you have inserted into living biological samples. This technique is primarily employed in the field of biology and neuroscience for selective disruption or modifying specific cellular structure or components without causing significant damage to surrounding tissue. Selectivity. Selectivity is the key word here. Either you disrupt, you burn it off, or you modify. Now, you might ask how it is going to get modified. So, the process is pretty simple. All these neural transmission, at the end of the day, they are biochemical reactions. Yes, even your transcription, translation, the central dogma part, these are biochemical reaction nonetheless. And every reaction has an optimal temperature where it performs the best. Hydrogen with oxygen will create water, no, everybody, but how much or how efficiently or how fast this process will occur depends on several environmental phenomena, one of them being temperature. So, every chemical reaction has some sort of a optimum temperature range at which this particular chemical reaction works with most efficiency. Using laser light, you can heat certain areas up. So, for example, if you see a loss of neurons, right, if you see a loss of neurons, there is just like your printed circuit board where a connection has been broken, a wire is cut off. What do? You replace with another wire. What do you do when similar thing you find out inside the brain that a neural connection is broken? A neural connection is broken. Now, you need to recreate the neuron, you need to connect it the neuron. Now, for a time being, several areas of the neuron, you know about glia, we have discussed about neuroglia, they contains the stem cells. These stem cells can convert themselves into neuronal cells and repair the damage sustained by the neurons, but you can aid this. You can aid this by selectively increasing or optimizing the temperature around that particular neuron, that particular tissue, which can results in development of specific tryptase molecule or those enzymes that helps in transcription or translations. You can help a particular temperature which will attract more amount of those tryptase or that enzymes that helps in translation or transcription from DNA to RNA, RNA to protein, and in turn accelerate accelerate the process of protein generation, which in turns will repair the tissue. Maybe it has so happened that the stem cells within that person, an old person's brain, has exhausted itself or is not working. So, you are simply giving it a boost by changing the temperature or even pH of the surrounding area. How the pH is changed? You create that bubble, remember that bubble from previous section, you create that bubble, the overall temperature, overall liquidity, overall presence, because you create an optoelectric field with this laser. The ions can be modified to come to specific areas, thereby the polarizability, the pH, etc. can also be modified. Obviously much more complicated than said, but the potential is here, and thereby you can help in not just breaking down something like a bad cell, bad tumor, bad neuron, but you can also modify and repair specific cellular structures or components without causing significant damage to surrounding tissue. So, that is overall the essence of nanosurgery. That is the overall essence of nanosurgery. So, principles and processes of multi-photon nanosurgery. Multi-photon nanosurgery capitalizes on the phenomenon of nonlinear optical interaction. When material is irradiated with high intensity laser light, multiple photons can be absorbed simultaneously within a specific volume, resulting in energy absorption sufficient for inducing a chemical or physical change in the material. So, you try to do this, a chemical change or a physical change. Physical change, burn it out, damage it because of the absorption. Absorption results in heat dissipation. Heat dissipation can basically burn it, or you can induce chemical changes into the material, just like what I discussed previously. A critical component of multi-photon nanosurgery is the precise focusing of laser beams. This is typically achieved using high quality lenses. The focused laser beam creates a highly localized region of high photon density, allowing for precise manipulation. If you have focused only in this part using your sophisticated lens, and these days you can break even the diffraction limit, meta lenses and whatnot, the photons are only activated in this particular region, and this dendrites and axons, these dendrites and these axons are no longer affected. This energy is absorbed by the molecules in the sample, leading to process such as multi-photon absorption and ionization. This ionization can result in formation of plasma and high energy state of matter composed of ion and free electrons. Plasma formation leads to chemical and physical changes within the targeted structure. Plasma, the fourth stage of matter, where electrons have generated sufficient energy, they are flying away from the nucleus is separate from the electrons. These electrons can in turn react with the surrounding molecules, mostly oxygen. Our body has huge amount of oxygen there, or they can react with the water presence, knocking out one of the hydrogen atoms out, thereby creating a hydroxyl group OH minus, or radical oxygen group, reactive oxygen species O double dot, radical oxygen O minus, and they are highly highly energetic. Reactive oxygen species, highly reactive. They can simply be used to eat up, they can simply be made oxygenation is by definition heating up or burning something. They can be utilized for eating up specific areas that you want to target, that you want to remove, but at a neuronal level. Very few surgeons can actually do an operation and take out a single cell. Using laser, perhaps they are more equipped to do that. The energy absorbed by molecules can result in bond breaking, fragmentation, or creation of reactive oxygen species ROS. These effects collectively causes disruption or ablation of the structure. The parameters for nanosurgery, including laser power and exposure time, are often adjusted through iterative process. Now, both previous lecture and this lecture, you have to understand they are still under development. Yeah, we are not simply putting lasers inside human brain yet, it's still to be standardized, but overall the potential is there. Researchers carefully optimize these parameters to achieve the desired level of disruption while minimizing damage to the surrounding area. Selectivity is the key. You cannot simply burn away entire brain tissues and then say that okay, the tumor is removed. You have to be very, very selective about what needs to be done, and these selectivity, this optimization, computer science or even electronics know how difficult optimization is, but we need to achieve optimization in this, which is still lacking. In mouse brain we are trying to do, and hopefully we will get this soon. So, lasers and nanosurgery regimes using femtosecond oscillator, very small pulses existing for few femtoseconds, so that you do not damage the nucleus, damage the atom. The damage is minimum, no matter how much localized your area is, you are going to cause damage, and sometimes you need the damage, but still you need the heat to be dissipated locally, closely and as soon as possible. Effect involved thermoelastically induced formation and growth of plasma bubbles. Mechanisms are related to thermoelastic effect. It absorbs, it swells up and then dissipates the heat and comes back to its original position. How fast this process works determine how successful your surgery and how localized your surgery would be. Low repetition rate regime. So, both high repetition and low repetition employs amplified series of pulses with energy slightly above optical breakdown point. Effects are mediated by quasi free electron induced decomposition and quasi free electron. Quasi free electron means they are almost free, but still have some amount of connection with the nucleus. They are not completely out of the bulk, just like your surface plasmons. They are almost free, but they are not leaving the bulk. Free electrons are generated to photoionization and impact ionization. They react with surrounding areas, ionize it, creates Ross hydroxyl groups and cause some kind of a chemical disruption. Nanosurgery relies on lasers and precise targeting methods to manipulate biological structure. Both high repetition rate and low repetition rates are used. The extent of damage depend on laser power, repetition rate and pulse duration. There are several advantages, the one being high precision, then you have minimal collateral damage or surrounding damage, non-invasive, or I think it is minimally invasive rather than non-invasive, real time versatility, you can target selective areas, you can even be clever and allow some exogenous fluorophores or exogenous material like nanoparticles to be attached at specific areas of the brain and then send the laser light specifically there to create this bubble, which will then in turn disrupt the laser. So, basically photodynamic therapy, photothermal therapy of the brain. Several applications, central nervous system studies, laser nanosurgery has been applied for mammalian CNS to study axon regeneration and synaptic competition. The technique has been used to disrupt CNS neurons. You can both repair as well as disrupt. Microvascular studies, laser induced lesions have been used to study microglial migration, astrocytes, focal photothrombosis and vessel ruptures models. These are, you remember those port wine stain areas where abnormal vesicles can be disrupted, but at a very, very nanoscale level. Multi-photon nanosurgery along with multi-photon microscopy can be used to mark specific cells for subsequent correlative light and electron microscopy experiments. You have application not just in neuroscience, but cancer treatment, gene editing, cellular repair as well as drug delivery. Researchers are investigating the use of nanosurgery to open cellular membranes temporarily, allowing for more effective drug delivery. Remember, we can open up certain areas we discussed in previous lectures, certain gates inside the cell membrane and then allow not just ions, but some drugs to go inside and then remove the laser so that the membrane closes, thereby the cell having more chance of retaining the drug. Cellular repair. So, repair, remember, is the far challenging one. Yes, it is pretty easy to disrupt, burn that area and damage that area and ablate that area, all of those things. But cellular repair, what do you do when the axons are no longer connected? You can, that repair part is the most complicated and the most challenging one, and using lasers and nanoparticles and quantum dots, if you are able to do it, you are very close in saving some human being's life, and that is overall the essence of nanobiphotonics, the aim of nanobiphotonics. Why are we doing any of this? This is simply to save human lives, right? Gene editing, optogenetics part is something that is coming up, and cancer treatment, nanosurgery to remove cancerous cells. So, in conclusion, offering nanoscale precision, the technique allows for targeted manipulation of individual cellular structure through localized plasma formation. Multi-photon nanosurgery is minimized collateral damage. The synergy of multi-photon nanosurgery with living imaging permits real-time observation, and from neurons to complex tissue, the technique is pretty versatile, right. So, these are the concepts that I covered today, and these are my references. Thank you very much.